Hello, everyone, and welcome. I'm the host of today's online symposium, Jessica, from GenScript ProBio Marketing Department. On behalf of GenScript ProBio, I'd like to thank you all for attending today's symposium. GenScript ProBio is a subsidiary of GenScript Biotech Corporation, proactively providing end-to-end CDMO service, from the drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in cell and gene therapy, vaccine, biologics discovery, and antibody protein drug to accelerate drug development for customers. This online symposium will include 4 parts. In part 1, we will share a 10-minute overview introduction of ProBox platform, and for the extra 3 parts, from upstream and downstream process development to analytical development, each part will take 30-minute presentation and 10-minute Q&A session. You can submit questions at any point during the symposium using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's first speaker, Dr. Ying Li, Director of Purification Process Development Department of GenScript ProBio. Dr. Li leads the CMC purification process development and provides technical leadership in new technology platform development. She joined GenScript ProBio in 2022, bringing over 10 years of biotechnology experience spanning multidisciplinary areas. Prior to joining GenScript ProBio, Dr. Li was an R&D manager in Merck KGaA from 2017 to 2022. Lead the development of viral vector manufacturing platform and biocontinuum platform. Dr. Li obtained her BS and PhD in chemical engineering from National University of Singapore and the Pennsylvania State University respectively. She is a frequent speaker at ACS and AICHE and has authored 8 publications in top-tier bioprocessing journals. For the first part, Dr. Li will share the overview of ProBox platform. Now, I'm delighted to hand over to Dr. Li. Dr. Li, please.
I hope you can all see my screen right now.
Not yet. Wait a minute. Okay, it's okay now.
All right. So let's get started. Hello, everyone. My name is Ying Li. It's my great pleasure to present today's topic, address CMC challenges with ProBio toolbox platform. Let's start with an overview of current global biologics market. In 2021, the global biologics market is projected to be worth over $330 billion, with a CAGR of 9.1% expected over the next decade, due to the broad application of biological drugs in a variety of indications. From the regional perspective, as you can see, North America still holds the largest market share in the global biologics market. And on the other hand, Asian Pacific is expected to be the fastest growing region in the global biologics market. In terms of the product segment, currently monoclonal antibodies segment still held the largest market share. However, there is a strong interest and trend towards the design of more advanced therapeutics to achieve improved efficacy through novel mechanism of action, including bispecific antibodies, antibody drug conjugates, recombinant proteins, cell and gene therapy, etc. ProBio, which is the CDMO segment of GenScript, has built a strong track record providing CMC services for all of these different biologics. This slide provides an overview of types of projects our platform has completed on record. As you can see, about 50% of our projects is related to mAbs, while around 25% is bi or tri-specific antibodies, with the rest of 25% related to fusion proteins and recombinant proteins. As we all know, these types of complex biological molecules pose significant challenges in the process development, from the upstream production to downstream purification and formulation. We have spent a lot of efforts in the past years to summarize lessons learned from the accomplished projects and developed our ProBox platform. Let's first take a closer look at the types of molecules collected in our ProBox platform. Starting from bispecific antibody, it could be broadly categorized into IgG-like or Fc-related bispecific antibody and non-IgG-like or Fab-related bispecific antibody. Under Fc-related bispecific antibodies, a typical structure includes symmetric bispecific antibodies, such as IgG fused with single-chain Fv or single-domain antibody. On the other hand, asymmetric bispecific antibodies, which include the classic structures, which could be assembled both in vitro and in vivo, and other different asymmetric structures. And here are some examples included in our non-IgG-like examples, such as Fab fragment or a single-chain Fv fragment fused with multiple single-chain Fv in the conjugation structure. On the protein side, we are proud of completing IND filing and GMP productions for a number of complex recombinant proteins. Here we categorize them into two different groups. The Ig fusion protein, which could be further divided into Fab fusion protein or Fc fusion protein. And in addition, quite a number of recombinant proteins have been studied in the past, including spike proteins, microglobulin, enzymes, or growth factors, etc. I would like to highlight the average titer and yield, which has been calculated for all the different molecules in the past. So for bispecific antibodies, the average titer achieved is up to 3.2 grams per liter under our platform, with average yield of 52%. And for fusion proteins, the average titer is 3.6 grams per liter, with average recovery of 44%. There are many unique manufacturing challenges posed for these complex molecules. For bispecific antibodies, for example, the generation of byproducts such as homodimer, aggregates, and fragments, those are difficult to be purified byproducts from the cell culture. And in addition, the low expression level and development of accurate analytical methods are also challenges faced for bispecific antibody development. On the recombinant protein side, in general, the low protein stability and complex PTM, the design of the capture steps, especially for proteins without any tag, removal of HTP, and of course, improving expression level and development of analytical methods such as bioassays are challenges faced in most of the recombinant protein projects. To address this problem, significant time and effort needs to be spent to identify effective solutions during process development, and sometimes the solutions may not be adequate. So here is an introduction of our ProBox process development toolbox. As I mentioned before, this is based on a lessons learned from over 100 ProBio CMC projects and integration of 200 challenges and solutions commonly faced in process development. And here is an overview of the ProBox. Can you still see my slide?
Thanks for Dr. Lee's introduction. Now, please let me introduce the second speaker, Mr. Rubin Zhang, Director of Nanjing Antibody PD and MSAT Department of GenScript ProBio. Mr. Zhang received his master's degree in biotechnology from Northeastern University in 2014. In the same year, he worked at Sanofi Genzyme as a process engineer for upstream process development of antibodies and recombinant proteins. From 2014 to 2020, he worked in the process development department of Merck Sharp and Dohme as a researcher and senior researcher, responsible for the upstream process development, process characterization, and validation, as well as leading the upstream process development of Merck Sharp and Dohme's continuous production and process online analysis upstream development. During this period, he participated in the publication of 7 academic articles. From mid-2020 to 2022, he worked in Hangzhou Yi'an as Deputy Director and Director, responsible for Upstream Process Development Department, in charge of CDMO, Upstream Process Development, and also promoted and enhanced the feedback process and continuous perfusion process platform. In this talk, Mr. Zhang will discuss ProBox solutions for upstream process development. Now, I'm delighted to hand over to Mr. Zhang. Mr. Zhang, please.
All right, thank you for Jessica's introduction, and thanks very much to Dr. Lee to have a brief introduction about the ProBio's technology platform. Now I'm going to talk about the cell line development and upstream process development toolbox for complex biologics. Here is an overview about the platform toolbox for the cell line and upstream. Our toolbox includes vector design, which including codon optimization, signal peptides, which signal peptide to choose, and the vector system for the bispecific antibodies. And also we have topics about transfection. We also have topics about clone selection, including how to do the single clone confirmation and what is the selection strategy for high titer and good quality clones. We also have a very good strategy to do the clone stability study, which can balancing the time for development and also the clone stability. For upstream, a lot of times the topics includes titer increase, quality attributes optimization, intensified fed batch, and perfusion cell culture. For upstream, for scale up, a lot of times we may have a lactate spike or low cell growth and a low QP problem when we scaling up the process. So these toolbox is comprehensive review of all these vectors. Today I'm going to choose some of the topics from the toolbox. So this is today's topics. First section is about the titer improvement. As we all know, a good cell line is always the foundation for success and high titer upstream. So I'm going to talk about our CHO genes and apt vector system from the GenScript ProBio. After that, I'm going to talk about the dual vector system for bispecific antibodies, which can greatly increase the productivity and also the purity. The third one I'm going to talk about is upstream parameter optimization, followed by intensified fed batch process and perfusion process. For N-glycan modification, which is another big part for upstream, I will talk about how to increase the galactosylation, how to decrease the fuel oscillation, and how to increase the cellulation for long half-life product. On the third big part, I'm going to talk about enzyme activity improvement, and last but not least, I'm going to talk about the scale-up issue. So for each topic, I will have solutions and case studies. Again, for the overview for cell line and the cell culture process development, when a new CMC project come in, it always start with the cell line construction. At this stage, we want to produce high productivity clones using CHO cell genes and a tighter up vector system. The next step is always clone selection. We're using 24 well plate for clone and Ember 15 for clone selection. In the same time, we also have a comprehensive strategy to guarantee the clone stability while balancing the development time. For media screening, GenScript ProBio have a top-tier media brands that are available for quick tests and get it ready for use in the process. For bioreactor process development, we would focus on the process optimization of temperature, pH, culture duration in 3 liters. In the same time, if our clients would need a higher titer, we would develop the intensified fed batch for the specific project. And if we have unstable product, we would recommend to use a perfusion process to develop the unstable products. When scaling up, ProBio have a proper scale-up strategy to guarantee the comparable performance between the pilot scale and the 3 liter. And also, ProBio have its own good GMP production system that can meet the quality requirement for both Europe, US, and mainland China. Now I'm going to talk about the CHO-K1 genes and up vector system. The CHO-K1 genes is GenScript ProBio's in-house host cell line. We have a commercial use license from ECACC, and the cell was adapted to serum-free suspension culture in ProBio. And coupled with the host cell line, we have our tighter up vector system. We insert the transcription active region. This vector can insert into the transcription active region of the chromosome. In the same time, it can speed up the stable pool stage, which can shrink the development timeline for cell line development. It can significantly improve the titer of difficult to express molecules, and we utilize this vector for map by specific antibody and protein productions. On the left bottom, you can see the difference between our classic vectors and the vectors using the up vector system. Different from the classic ones, we actually have a quite high producing clones from the beginning. And in the middle of the graph, you can tell that we not only have 1 or 2 high producing clones, but each transfection using the vectors new vector system can harvest multiple high producing clones from the transfection. This actually provides a solid foundation for high producing upstream process. The up vector system was start in service starting from the end of 2022. So we're still accumulating data, but now I want to review some of the data from our CHO-K1 genes host cell line coupled with our classic vector system. On the left bottom, we can see the average titer for different protein modalities. For monoclonal antibody, on average, we have 5.4 gram per liter. And for bispecific antibody, like what Dr. Lee mentioned, the average titer is 3.2 gram per liter. And for recombinant protein, the average titer is 3.6 gram per liter. And in terms of the distribution on the right figure, for monoclonal antibody, 10% of the project will fall in between 2 to 4 grams per liter, and 40% of the product will fall in the category between 4 to 5 grams per liter, and 40% of the project will fall in the category between 5 to 8 grams per liter. And luckily, we have a more than 10% about the project can reach more than 8 grams per liter, even without any cell culture optimization. The next question after the cell line development is naturally goes to the stability of the cell line. ProBio guarantees more than 60 PDL stability for the project from the TROK1 genes and the TROK1 ADCC platform, and we guarantee there's no sequence variation in genetic level. How much is PDL 60? Would PDL 60 enough for the large scale? The figure in the middle actually answers this question. We set the primary cell banking as a PDL 0, and for master cell bank, the PDL is 12, and for working cell bank, the PDL is 24. After we saw a while and start to scale up, if we scale up to 200 liter, the PDL will be 40, and at the end stage of 2,000 liter, the PDL will be 43, and at the end stage of 8,000 liter, the PDL is 46. You can see there is still a long way to go between the PDL number we guaranteed. In the same time, our host cell line have excellent run-to-run comparability. On the bottom figure, I'm showing one example that we have a continuous 3 runs in line, and each run all the runs have a similar viability, similar titer, and a similar product quality. Let's switch gear, talk about a little bit about bispecific vector system. The reason I mention it is because for bispecific manufacturing, we always have some monospecific impurities. Sometimes when we have a really low product yields and high aggregation concentrations, and some of the undesirable fragments. To solve that problem, ProBio actually developed a 2-vector system to improve the productivity and the purity for the bispecific products. The difference, like what I showed on the left bottom, instead of using 2-vector, we're actually using 2-vector system, and for 2 chains, we separately copied them into different vector. Then we adjust the vector ratio before the formal transfection. The first transfection is more like a try to find out the best ratio between different vectors. Then we do the formal transfections according to the vector ratio we learned from the last run. By using this strategy, we can greatly decrease the homodimer and increase the heterodimer productivity and purity. This is a real case from our projects. On the top, I'm showing the vector system using the single vectors. From the SDS page, you can tell our target product is chain A plus chain B. However, quite some of our product from this transfection are chain 2, chain B, or only chain B alone. And this data further verified by the SEC HPLC and NRC SDS, and the cell poop fed batch production titer was only 0.5 gram per liter. On the bottom is the 2-vector system transfection after the ratio optimization. You can see from the SDS page, the majority of the product are the correct form, and this number is also verified by the SEC and NRC SDS. If we analyze the case on the top, you can tell there's a large amount of chain B compared with the chain A expression. Because of that, during the transfection in the transfection of the dual vector system, we would greatly increase the vector number for the vector concentration for chain A, but decrease the vector concentration for chain B. So eventually we can balancing the number of the chains of the two chains produced. By doing that, the cell pool production titer was increased from 0.5 gram per liter to 5.4 gram per liter. After the cell line, I'm going to talk about the upstream process development. During upstream, how could we increase the titer? The way including do media screening and feeding strategy development. Also, sometimes additives would help for the cell culture performance. In terms of the process parameters, we can always optimize temperature, pH, and even DO level. If the common measures wouldn't help for the titer, we can also develop perfusion process or intensified fed batch process if we want to greatly increase the titer. Here is a good example about temperature optimization enhance the productivity. Internally, this data was organized from the 11 projects. There are literature saying behind the product temperature decreasing, there's a literature explaining the principle behind it. So after the temperature shift from 37 to lower temperature, 33 degree or 30 degree, the lactate metabolite was greatly optimized. And also the cell cycle was arrested at G1 phase, which stopped the cell from growing, but changed the cell from growing to production. In the same time, the metabolic was also changed from lactate generation manner to a lactate consumption way. The lactate would go into the TCA cycle and also phosphate pathway and also glycolysis cycle. By doing that, especially for low expression molecules, the titer was greatly increased. On the bottom right, you can see for MAPS, after the temperature optimization, the titer can increase to more than 30%. And for non-MAP product, a lot of times it can greatly increase to 172%. This is another great example of the downshift can increase the productivity in the same by enhancing the productivity and also increase the duration of the cell culture. This case is from SCFV expression. This molecule itself have really low expression in our platform process. However, we start process development with 3 different temperature. On the left bottom, you can see at 30 degrees, the cell only reached 15 million cells at peak cell density. And at 33 degree, it reached around 18 million cells, and at 37, it reached 20 million cells. However, as the temperature goes up, the viability actually drops faster. So at the 30 degrees, the viability can maintain around above 80% on day 15. If we look at the titer data on the right, for titer, the 30 degree titer actually increased more than around 6-fold compared with the culture which kept at the 37 degree. And in terms of the QP was also greatly increased comparing with the default 37 degree process. Since at the low temperature, we also have a really good viability. So to further increase the titer, this process was the process duration was extended from 15-day process to a 17 days process. On the top middle, you can see the product quality attributes. Starting from the day 15 and day 17, the product quality attributes are quite similar. However, starting from the day 19, the product quality starts to getting worse. So the process team decided to stop at day 17 as a final process. This process was scaled up to 250 liter, and after the 3 runs was quite comparable with different scales. Finally, before the before the process optimization, the titer was only 0.3 gram per liter, and after the process optimization, we successfully increased the titer to 2.06 gram per liter. This is a good case for high feeding increased titer. So this is the IgG4 case. During the platform process, the viability was fine and the titer was fine. However, for our platform, as a platform process, we also test a different feeding strategy. For high feeding strategy, which have around 30% of the feeding volume compared with the basal media, it increased the average titer from 6 gram per liter to 8 gram per liter, even though there's no impact to the cell growth. As a result, by increasing the feed volume, we successfully increased the productivity from 6 to 8, and there was no influence on cell growth for IgG4. If we want a significantly increase for productivity, the common measures may not work, so we would use intensified fed batch to greatly increase the titer. Different from the conventional fed batch process, at N-1 stage, we will use perfusion. By doing this, we can greatly increase the inoculation cell density from 0.5 million cells to 10 or even more 20 million cells per ml from day 0. And this will shorten the production cycle. In the same time, it will greatly increase the titer. The figure in the middle is actually highlights the different 14 projects that use the intensified fed batch. On average, the production titer was increased more than twofold, and in some cases, it's increased more than threefold or even reached the fivefold. Here I'm using a FC fusion protein case. This is a FC fusion protein, and it has a high concentration formulation, which require high titer. So we start process development in both fed batch mode and the intensified fed batch mode. For fed batch mode, the inoculation cell density was 0.5 million cells per ml, and for intensified fed batch, the inoculation cell density was 10 million cells. From the figure in the middle, you can tell the titer was increased from on average 6 gram per liter to more than 10 gram per liter. And after the process optimization, the highest titer reached 15 gram per liter. On the right bottom, you can tell there's no quality attributes difference between the fed batch process and the intensified fed batch process. Another way to increase the productivity is using the steady state perfusion process. Here, I'm using a showcase of the IgG1. This culture duration was up to 84 days. The productivity was about 28 times compared with the traditional fed batch. In traditional fed batch, the productivity was 4 gram per liter. And at the end of the 84 days, the accumulated titer for perfusion process was 110 gram per liter. And the quality attributes was stable during the steady state phase for this perfusion process. Here I'm showing a real case for a FC fusion SCFV case. For this project, 2 SCFV was added to the bottom of the protein. And this product suffers from the low expression level, and also in fed batch process, after the one-step purification, there was still very high HCP remaining in the process. So the process was developed and to a perfusion process up to 27 days, and the productivity at the end of the day was increased by 10 times. For fed batch process, the titer was 0.78 gram per liter, and for perfusion process, the cumulative titer was 7.7 gram per liter. On the bottom left is showing the cell growth profile, and on the middle of the bottom is showing the productive activity profile, and on the right bottom is actually showing the greatly decreased host cell protein number. For intensified fed batch, after protein A, there are still around 3,000 HCP. However, for the perfusion process, the number was greatly reduced to 400, and for drug substance, it was further reduced to 20 ppm. And glycan is also one big part for upstream for the product quality attributes modification. There are common requirements like fuel constellation modification, galactose modification, and the salicylic acid modification. Here I'm showing a case, an IgG4 case to increase the galactosylation modification. By adding the galactosylation modification, we can improve the binding activity. That's why the clients would need to increase the galactosylation. There was several components was tested, including some commercial available reagents and also literature available components. After the try, galactosylation was increased from 3% to 28% with a mixture of trace elements, nucleoside, and also the substrate. There was no significant influence on titer CX and also MEN5. For the principle behind it is very easy to understand. So galactosylation is also an enzyme activity. In order to optimize this enzyme activity, we actually find three factors that actually can help. The first is the substrate of the concentration of the substrate. Therefore, we add more galactose into this media. In the same time, there are always cofactors for the enzyme, including some of the trace element and also some nucleotide. That's why we added them in. And eventually, the galactosylation level was increased to around 30%. Well, it has no impact to other N-glycan profile, especially the MEN5 was kept low. The other part I want to talk about the fuel constellation reduction. Clients may have needs to increase ADCC activity. There are actually two ways to, in order to increase ADCC activity, so fuel constellation level need to be bring down. There are two ways to do that. The first one is the best one is always to modify the cell line directly. At ProBio, we actually developed the one cell line that can knock out the FU8 gene to produce afucosylation antibodies. The FUT8 enzyme is actually responsible to adding the fucosylation to the protein. And after we knocked out that enzyme, fucosylation cannot be added to the protein. And the cell line we actually have it is CHOKI1 ADCC plus cell line is a stable cell line for CMC service with a comparable productivity and also clone stability. On the right, you can see the fuel constellation level using the ADCC enhanced cell line compared with other commercial available additives to decrease the fuel constellation. For the best additives from the commercial available reagent, it can decrease the fuel constellation to 35.1%. However, for ADCC enhanced cell line, there's no fuel constellation can be detected at all. However, for the clients already have their cell line, there's no way that we can modify that host cell line in short period of time. The other strategy is to use the additives to decrease the fuel constellation. This is a good example for a IgG1 project. So additives were added to the cell culture medium, and the fuel constellation content decreased from 98% to 35%. There was no influence on titer and other quality attributes, including MEN5. On the bottom, you can see the MEN5 content was constant between different conditions, and the titer was comparable for the additives B and the control. And by doing that, the fuel constellation, the ADCC activity would actually increased more than tenfold by decreasing the fuel constellation. In an interleukin projects, we actually add additives to raise the glycosylation. When there's no glycosylation in the interleukin, the protein becomes unstable. If you look at the right up figure, on the one doesn't have Na there, that means that specific molecule don't have any N-glycan there. And after 40 degrees for 2 weeks, that product actually quickly degraded to 3 different main peaks. And for the ones with N-glycan on the top, they are stable for 2 weeks at 40 degrees. And the glycosylated interleukin showed much higher stability. And also, it was reported that saturation prolonged the half-life of the protein, like the interleukin. So for this project, we're looking forward to increase the glycosylation in the same time, increase the saturation as well. Again, different components, additives were adding to the basal media in order to increase the glycosylation. And we have trace elements, nucleoside, and also the substrate were adding to the medium. The saturation was successfully increased from 42% to 90%, and glycosylation was increased from 80% to 98%. And most of the interleukin were glycolized, and the non-glycolized protein was not even detectable. After optimization, if you look at the upper right figure, you can see all of those has N-glycan on them, and in the same time, which is more important, is the 3SG, the NAL, NAC 3SG peak becomes much larger. That means the saturation content will also greatly increased, which will help the half-life. Let's switch gear, talk about the enzyme activity. So we have one project that is a lysosome enzyme case. So for this enzyme, it needs to be modified between release to the environment. Otherwise, this enzyme will not be active. So during our upstream process development, we find out as the duration goes up, and also as the titer goes up, the specific activity of this enzyme actually goes low. So it has a reverse trend of cell culture duration and the specific activity of the self-taste. Later on, so we developed a perfusion process in order to increase the specific activity of this product. On the upper right figure, fed batch process and perfusion process was compared. For perfusion process, the cell density was reached 60 million cells. And for the specific activity, the activity was increased from 30 to 300, which is a tenfold increase just by switching from fed batch process to a perfusion process. In order to further increase the activity, we also find out the cofactor of the enzyme. The two trace elements were also added to the cell culture, and the concentration of these two trace elements were determined using a DOE study. And in the bottom middle, you can see the activity was increased in the DOE studies. Some of the conditions actually reached 900. And so at the end of this process, the enzyme activity was increased about 30 times after the cell culture optimization by switching the fed batch to a perfusion process and also adding the cofactor trace elements into the media. The last but not least, I'm going to talk about a little bit about the scale up. For a lot of our process, if the cell line is from non-platform cell line from the clients, then scale up strategy need to be optimized when running the cells. Because when scale up, depending on the metabolism activity of the cell, some of the cell may more sensitive to the environment, and we may see the lactate spike during the process. On the left bottom is a real case happening in our pilot plant. The lactate spiked in the middle of the process. And in the following run, we actually mitigate the issue by adjusting the agitation, DO, and the macro sparge. And the lactate metabolism came back to historical trend in following batches. For scale up, there are several factors we also wanted to consider. One of the most important thing might be the DO level, and also nowadays, more and more cells have a high cell density, which reaches 30 millions or even 40 millions. So the P188 concentration is also a good factor to optimize for our process. That's all I have for today. Now I'm available for questions. Thank you a lot.
Thanks for Mr. Zhang's excellent presentation. If you have any questions, please type it on the Q&A panel. As you enter the questions, please also follow our LinkedIn account, GenScript ProBio. As you enter the questions, please also follow our LinkedIn account. There will be more online sharing resources, and we will share them on our LinkedIn at the first time. Thank you. Okay, we received some questions from the audience. Mr. Zhang, could you please answer this question? Would IFB significantly increase the development timeline for IND projects? If yes, how long time is expected to expand the development time frame? Could you please answer this question?
Yes. Thank you for asking this question. So indeed, for a lot of our IND projects are very time sensitive. And for the monoclonal antibody, we promise our clients can get their tox batch material by the end of month 6. And for a bispecific antibody, it takes 1 more month for a fed batch process. However, for intensified fed batch, we also have to optimize the feeding strategy and the time optimization, temperature optimization. So therefore, a lot of times 1 more month is needed. So yes, intensified batch would increase the development time for IND process. However, it will only have a 1 month increase in terms of the total time frame, which is not wouldn't significantly impact the IND process progress. Thank you.
Thank you. Thank you for Mr. Zhang's answer. And the next question is, for quality attributes adjustment, what to do solve the purity-related quality attributes, CX profile, SC, SEC, MEN5, and glycan profile.
Yes, product quality attributes is one of most very important consideration during the IND process development. And for us, we have our Trojan's host cell line, which is well trained. So majority of the time, from the cells, it produces the quality at the product produced from this cell have a right product quality attributes, which means the SEC a lot of times is more than 90% of the purity is the May peak. In the same times, the MEN5 is always below 10%. And for CX profile, the May peak of the CX profile is always more than around 60 or more than 70% a lot of times. This is because our host cell line is well trained, so we can get the product quality attributes right at the first from the beginning. However, if sometimes we may have the cell line from a customer, for those cell line we may consider to use different pH or different temperature shift, and also media screening in order to optimize the product quality attributes.
Okay, that's great. Thank you. Yeah. Thank you. And the next question is, what is the general large-scale tech transfer consideration if the process is transferring to bioreactors from different bands?
So for the for CMO projects, clients may have a different brand of the bioreactors compared to us in their IND process. When they're transferring to us, they may have this kinds of concern. However, it's not a big issue to ProBio because we have a comprehensive scale-up strategy. During the technology transfer, the first thing we always do is repeat the process in our small-scale process. And after that, once this process is successfully repeated in our small scale, then it becomes to, in our system, how to scale up this process from 3 liter to 500 liter or 200 liter in our own system. In that case, there would be no problem like transferring a pilot scale process from different brand of the bioreactor.
Okay, thank you. Is there any question in your Q&A panel? If no more question, let's move to the next part. Okay, now please let me invite Dr. Li again for the third part sharing. In this talk, Dr. Li will discuss ProBox solutions for downstream process development. Let's welcome Dr. Li.
Thank you, Jessica.
You're welcome.
I hope you can see my screen right now.
Yeah, it's okay.
All right. So hello, everybody again. So under this session, I'm going to talk about downstream process development toolbox for complex biologics. So first, let's start with an overview of common challenges faced for purification process. So starting from depth filtration, there are a number of problems related to antibody reduction or protein absorption, and followed by capture step. And in general, the capture of antibody without Fc fraction or protein without tag could be problematic to design a capture step. And in particular, some unstable proteins may be difficult to be captured or eluted, especially at low pH elution conditions. And similarly for VIN, for proteins unstable at low pH or unstable in SND, we need to identify strategies to develop this VIN step. And in addition, for pollution chromatography steps, typically including a number of ion exchange, CEX, AEX, and hydrophobic interaction chromatography, and mixed mode chromatography, the removal of some difficult product-related impurities, such as aggregates or fragments, could be a big challenge. And for some antibodies, and in particular some proteins, the removal of difficult HCP is a big challenge. And well, at the same time, to improve the overall product recovery. For nanofiltration, in general, the low loading capacity and low stability proteins are common concerns for this particular unit operation. And finally, for UFDF, the high concentration formulation pose significant challenge to the design of this unit. And here is an overview of the downstream toolbox. So here we covered quite a variety of different problems, as I just talked in the last slide. So we find, in general, this is a quite effective way to improve the development capability and shorten the development timeline. So here is an overview of today's topics. I'm going to select a number of problems that most people have expressed concern with purification and formulation for biologics, which include reduction, capture, how to deal with aggregates, homodimer, remove of HCP, and finally, high concentration formulation development. So let's start with the first challenge, the reduction of antibodies or proteins. So here is a case study we have accomplished in the past. So this is a fusion protein with isoelectric point of 7.8. And upon development, we identified this molecule is quite unstable upon storage in the room temperature when we first harvest the supernatant. And here you can see from this graph, upon harvest, the mean peak, the CE purity is up to 98%. Well, when the supernatant was stored at room temperature for 7 hours, there was a significant generation of antibody fragments, including the 2 heavy chain, 1 light chain, or some other impurities. And the mean peak purity dropped to 56.7%. Therefore, this molecule has high reduction tendency. And increasingly, we have seen a lot of cases like this, even for monoclonal antibodies. So in general, the antibody disulfide bond reduction is essentially a redox reaction that involves redox enzyme. So this particular reaction needs to be categorized by thioendoxin reductase, and also requires TRX, and also requires the NADPH to provide electrons to kick off the reaction. Therefore, there are a couple of methods based on this mechanism to reduce the reaction and reduce the reduction of the molecule, which include the use of microions or chelating agents, which will inhibit the redox reaction by reduce the reactivity of the enzymes. And oxygen or some other oxidants could consume the NADPH through a competitive reaction pathway, therefore to shut down the reaction as well. And in addition, minimize the cell lysis, therefore reduce the generation of the enzyme, lower the temperature, and pH can also slow down the reduction reaction. And our team have built a reduction evaluation platform to systematically evaluate and mitigate the risk of reduction. So under this case, we applied a few different strategies, which include continuous air sputtering for overnight, adding trace amount of copper ions, and or adding low concentration of histamine. And you can see from the results in comparison to the control case, there was a significant reduction of the generation of the 2 heavy chain 1 light chain impurities by using all of these different strategies. And finally, we selected increasing dissolved oxygen method because this is something very easy to be implemented in the large-scale manufacturing process. Next challenge is about capture. Like I mentioned, in general, the capture of antibody fragments without Fc fragment, and also the capture of recombinant proteins without any tags would be quite challenging to be done. And for the first case is related to a Fab fragment with a PI of 8.7. And here, this is integral Fab molecule. And for this particular case, our team screened different binding domains using different affinity chromatography resins. In particular, for Fab, there are 3 different domains we can potentially utilize, which include the VH domain, the VL domain, and CH1 domain. And there were a number of resins available in the market that we could potentially select from. And upon screening, we identified that for most of the Fab molecules, utilizing VL domain is the most effective way to capture the Fab fragments, and in particular with much higher capacity compared with using other binding resins. And in comparison, we screened a number of different capital L or protein L resins and identified an optimal one with a significantly improved overall capacity, which again reduced the overall cost for the manufacturer. And through this one-step capture step, we successfully achieved SEC purity above 99.5% with over 96% of C purity. So lessons learned is always select the best affinity resin for the right binding domain, and also screen different resins to get the highest capacity. And here is another case, which is a nanobody CHH fragment fused with an enzyme. So there were a number of challenges associated with this molecule. So first, again, this molecule does not contain any Fc fragment, so this is difficult to be captured. And in addition, because of the enzyme fraction, this molecule is quite unstable, so it tends to precipitate when pH drops to below 7.0. And this could be seen in this chromatography graph when we elute the bond products with using the normal acetate buffer at pH 3.5. We found the majority of the products were aggregates of fragments, and when adjusted to pH 7, the pH adjustment did not resolve aggregates, and the precipitation still exists. So for this particular case, first we did a comprehensive screening study to identify the optimal VHH binding resin, which was selected as our platform IC resin for all the nanobody related products. And after that, we also did an optimization for the elution process. In particular, we changed the elution buffer from acetate to glycine HCl at the same pH. So in general, it will still be able to elute the target protein while the eluent was at much higher pH, close to neutral at pH 7.2. So here it avoided the pH adjustment across the PI and also storing the protein at low pH, therefore results in a highly purified product with SEC purity above 97%. Next, let's talk about the capture for no-tag recombinant proteins. So for this particular case, we're dealing with a giant protein molecule, which is a tetrameric protein, and also at the same time at pretty low PI at 5.9, and this is not uncommon for a lot of recombinant proteins at acidic PI. So the challenges include, again, first, you cannot find any affinity reagents, in particular for this type of recombinant proteins. And again, this protein is highly unstable. So especially with the screening study to identify a design space for keeping the molecule to be stable, and identify that under low conductivity and low pH, the protein tends to be form precipitation, and therefore cannot be further processed without a product loss. And in addition, without effective affinity chromatography step, the HCP level is quite high. And therefore, to solve this problem, we first identify the right capture chromatography to capture this recombinant protein molecule. We end up with a CEX. This is in general our recommendation because utilizing AEX for the first capture step, sometimes you will end up with capturing the majority of pigments, which will result in blocking of your chromatography column. For utilizing the CEX, here we need to balance the stability and the recovery to choose the best loading conditions. Remember that we just mentioned there is a very narrow design space for this type of recombinant protein. And after that, we need to investigate the different pH and conductivity sensitivity between HCP and target protein, and we choose pH dilution in this particular case, which was effective in removing the target protein, diluting the target protein while removing the majority of HCP as well. In addition, a wash 2 step at pH 6.2 was added to further wash out the HCP, and arginine is added to the dilution buffer to eliminate the abnormal pH rise and further reduce HCP. As you can see, upon all this optimization with the wash 2 step and arginine addition on the elution step, the overall recovery is up to 92% with 88% of ICC purity and significantly reduced HCP level. So the next challenge is about aggregates. This is one of the biggest challenge in a lot of bispecific antibody development. So in this particular case, I include the case for the IgG fused with single chain FV because of the exposure of the hydrophobic region of the single chain FV. This is not uncommon to observe a significant percentage of high molecular weight present in the intermediate, supernatant and intermediate products. So for this particular case, as you can see, after the affinity step, there was still 8% of the high molecular weight, which was quite challenging to be removed. And upon optimizing AC, including the wash and elution step, or the CEX step, cannot remove aggregates effectively. So under this situation, we generally recommend using a mixed mode chromatography. So for this particular case, a mixed mode AEX in flow through mode was utilized for high molecular weight removal. And we adjust the optimal loading pH and conductivity to achieve the best separation of mono monomer and the high molecular weight. With one-step chromatography, will achieve a final ICC purity above 98%. Again, in our experience, utilizing multiple interactions, including hydrophobic interactions, electronic interactions, or hydrogen bonding, will significantly improve the separation for some difficult to be removed impurities, such as aggregates. So second case is also a bispecific antibody. So this case is an asymmetric bispecific antibody. Again, this molecule was designed in a knob-into-hole structure to prevent mismatch. And unfortunately, there was significant high molecular weight present in the AC alloys, around 20%. And we tried using different CEX resins, and also at the same time, some mixed mode CEX as well. And none of them achieve a pretty good separation of the aggregates and the MNP. At the end of the day, we utilize the CHT, which is a ceramic hydroxyapatite chromatography, which is a crystalline mantle of calcium and phosphate, which contains a calcium site with high metal affinity, and phosphate site, which is a classic CEX mechanism. In general, this chromatography is quite effective in removing a lot of high molecular weight. And there are a lot of tips to optimize this particular chromatography step, including the maintaining operation pH at a relatively high pH to extend the lifetime of the resin, and make sure to include phosphate in our samples and buffers at a base concentration to improve the resolution of the separation, and combine the elution with both phosphate buffer and sodium chloride buffer to get the best result. Again, some other tips include, again, of course, adding wash 2 to remove repeat fragment to improve the separation of the monomer and high molecular weight. So here, as the results shows that upon using one step of CHT, the high molecular weight drops from over 16% to less than 1% in this one single chromatography step, which is quite effective. And next challenge, again, which is highly relevant to bispecific antibody, is the removal of homodimer. Ruben just talked about reducing the expression of homodimer from the optimization using the viral vectors, especially adjusting the different ratios of viral vectors. From downstream perspective, in general, some of the mismatch of homodimers could be removed by some of the chromatography steps. So here is one example of asymmetric bispecific antibody. Even though a knob-into-hole structure was utilized, there was still significant amount of homodimer, the whole homodimer expressed in the cell culture, and it is up to 40%. And this is a quite big challenge for downstream purification, especially considering the similar chemical and physical structure normally observed for the two chains. And the capture and intermediate purification are not effective in homodimer removal. So for in this case, we selected mixed mode chromatography MMC for homodimer removal by both the charge and hydrophobicity difference. So here, upon again utilizing MMC, there was almost all the 40% of the homodimer was successfully removed with final purity about 99% in the final alloy, as you can see in the graph. One of the key for the success for this particular case is the relatively big difference in terms of the PI between the two chains. Upon analysis from IC-IEF, we could identify the delta pH between chain 1 and chain 2 is close to 1, which is considered to be pretty significant. Therefore, it is quite effective utilizing MMC or cation exchange mechanism to remove the two different mismatch species. And for HTP removal, this has been a big challenge for a lot of complex biologics, in particular for recombinant proteins. Because many recombinant proteins have relatively low PI, which is close to the PI range of HTP, making the removal of HTP difficult. And at the same time, there is no existing affinity resin for capture of many recombinant proteins, therefore leaving high level of HTP post-capture step. Oftentimes, this level is above 10,000 ppm. And on top of that, as I mentioned before, the proteins are generally unstable under certain pH and salt conditions, therefore further limiting the operation space for most chromatography steps. So in general, the solution, again, could start from upstream. So in our platform, we always select the optimal host cell system. In the ProBio system, in general, for the majority of the molecules, the starting HTP level upon capture or affinity step is below 2000 ppm. So that gave us a pretty good starting point. And again, the controlled cell culture process, including duration, VCV, and viability of the cell culture, could also help resolving some of the starting HTP level for downstream purification. So from downstream perspective, we have developed strategies to optimize the wash and elution conditions for a number of chromatography steps to effectively remove HTP species. And in general, we recommend using the hydrophobic interaction chromatography utilizing hydrophobicity difference to remove those HTPs, such as using HYC or MMC chromatography. And finally, most of the time, we will use intermediate DF step, which utilize the filter, optimize the filter and loading conditions to significantly absorb a lot of HTP species. Here is one example of Fc fusion protein. This is actually a factor VIII blood factor, which was again expressed with high HTP level and quite unstable molecule. So here is an overall path for removing the HTP level from over 60,000 ppm down to less than 20 ppm through four-step chromatography, three-step chromatography step process. So starting from the AC, the starting HTP level was quite high, and after that, the utilization of HYC was quite effective because part of the HTP with stronger binding ability will be removed from this particular step. And we compared two different routes going through the AEX directly or adding an intermediate DF depth filtration step, and identified that the AEX effectivity could also be significantly improved by adding intermediate DF to reduce the starting HTP level, which brings down the final HTP level to less than 20 ppm. And finally, this high concentration formulation is again one of the biggest challenge in the entire downstream purification downstream process, and including both purification step and formulation step. There were a number of challenges associated with high concentration DS process. The pressure drop was typically much more significant compared with lower formulation conditions. And generally, the yield of the process at UFDF step could be impacted. And sometimes a pH excipient drift could be observed due to the high concentration of protein expressed in the final formulation buffer. And of course, the formation of protein aggregation, especially under the conditions when the proteins are not stable. So there were a number of underlying root cause for this problem, including the high viscosity of the solution, the dominant or volume exclusion effect, and the self-association with the protein molecules. And all underlying that was highlighting the intermolecular interaction of the protein molecules, especially under high concentration. So here, a number of cases, we successfully developed strategies to address the challenges in high concentration formulation. The first case is a bispecific antibody with PI of 8.5. So for this case, the DS concentration is required over 120 milligrams per milliliter. And we observe a pH drift at higher protein concentration as seen in this graph. So the target pH, final formulation pH is around 6.5. But when we add the proteins, exchange proteins into target buffer, the pH was drift significantly above the target value. And therefore, we developed a strategic platform to compensate the effect of the pH drift and identified a different buffer for the buffer exchange step in order to compensate the pH drift effect. And also the increased viscosity results in the pressure challenges. And in general, you need to take a closer look at the optimization of the TFF step before and after the change of the buffer and at different protein concentrations. So in this particular case, the optimal operation flux, feed flux was also switched when the protein concentration was increased and the dye filtration buffer was switched from the initial buffer. And also for this particular case with the fusion protein, and again with low PI acidic protein, the DI's concentration requirement was over 140 milligrams per milliliter. And in particular, the overall concentration step, the concentration of the protein reached about 200 milligrams per milliliter. The increased viscosity, of course, will result in a lot of pressure challenges. So in this case, our team selected the optimal viscosity reducer in order to reduce the viscosity of the protein formulation. Here, as you can see from the table, adding sodium chloride was quite effective in reducing the viscosity of the formulation. As you can see, at high sodium chloride concentration, even for very high protein concentration at about 150 milligrams per milliliter, the viscosity level was below, was at 11 CP, which was much easier to be processed in the UFDF step. Therefore, in general, it is recommended to select and optimize the viscosity reducer and adjusting the concentration of the excipients to reduce the concentration, the viscosity of over concentration step. and of course, adjusting the inlet flow pressure and appropriately select the membrane cassette with in particular for the overconcentration step. So in summary, hopefully I have demonstrated today there were a number of unique challenges posed for complex biologics, such as bispecific antibodies and recombinant proteins that require significant time and efforts to search for effective solutions. And here at ProBio, we have developed a comprehensive platform to address typical challenges faced in both purification and formulation unit operations. And with that, I would be more than happy to take any questions you may have. Thank you.
Okay, thank you for Dr. Lee's excellent presentation. There are really many great case studies. Okay. If you have any questions, please type it on the chat or Q&A panel. As you enter the questions, please also follow our LinkedIn account, GenScript ProBio. There will be more online sharing resources, and we will show them on our LinkedIn at the first time. Thank you. Okay, we've got some questions, and the first question is, Dr. Lee, how to design a robust process with a good VCS performance while the target protein PI is low?
Okay, thanks for the question. I assume VCS means viral clearance study. And this is a good question. This is some challenge we have faced in the past projects, especially for proteins with low PI. So first of all, you need to make sure that in terms of viral clearance, regardless of its PI, make sure that the robustness of your nanofiltration and VIN step could still be available, because those are the two major effective steps to remove virus. And for those proteins which are unstable at low pH, make sure you select the best SC platform to address the VIN step. And second, of course, the low PI protein, typically, sometimes it's challenging to design a flow-through AEX unit. So to compensate that, typically we will need to take considerations of optimization of such as AC and CEX step, including adding some wash steps to more effectively remove the virus, and also considering there were a number of new products available in the market which could allow the operation of AEX at wider pH and a higher concentration, which could also be considered to be utilized in your case. So then for chromatography, you need to take a more careful examination in order to potentially compensate the loss of the viral reduction from potentially a flow-through AEX step.
Okay, thank you. Thank you for your question. And the next question is, based on your platform's experience, what insights can be provided for the drug developability of different types of bispecific antibody formats?
Okay, thanks for the question. I think the general principle is for all the different formats of bispecific molecules we have worked on, we find in general, the less modification, the more nature of the format it is, the better stability it is for the molecule. So for example, if you could choose a nanobody to fuse into a IgG, it is always better than fusing a bigger fragment such as single chain SV or a Fab fragment. And of course, in general, for most of the IgG related bispecific antibodies, our platform could address the majority of the CMC challenge, even though in some cases, some developability factors may not be ideal. For example, there's a tendency to form some aggregation levels or fragments. So for those cases, and in terms of developability, it is not ideal. However, this is something easier to be resolved through the CMC platform. However, for some other factors, for example, for some bispecific antibodies with pretty low thermal stability or pretty bad tendency to form aggregates, even at very low concentration. So for those cases, if we identify those tendencies, sometimes maybe it is better to modify the structure of the bispecific antibodies to improve the developability. We have quite a number of cases improving a different bispecific antibody format, including the symmetric ones and asymmetric ones to reduce aggregates and improve reduce the number of mismatch, which we could possibly share in the future talks. Thank you.
Okay, thank you for your introduction. And another question is, what are the differences in impurity release standard between recombinant proteins and antibodies?
Okay, I think this is quite good bridging question for the next session, but I will briefly cover this here. So in general, for recombinant for proteins, we have slightly different release standards compared with monoclonal antibodies. For example, in terms of the SEC purity, we could set a slightly lower standard at 90% compared with, in general, 95% for monoclonal antibody. Same for CESDS. Typically, we can set at about 90% in comparison to at least about 92% for IgG. And there are some differences in terms of the residue levels as well. For example, for the HCP, for a monoclonal antibody, it's typically below 100 ppm. Well, for proteins, especially for those recombinant proteins that are difficult to remove HCPs, we could potentially set the level at 500 to 1000 ppm. And for HCD, typically for proteins, it's down to 10 nanograms per dose compared to 100 picograms per dose for monoclonal antibody. So those are the differences that I could come up with for now. Thank you.
Okay, thank you. And here is a question may not very related to your talk, but could you please introduce the approximate time for biosimilar manufacturing? Do you know this question?
Yes. We have worked on a number of biosimilar cases. So in general, it took us around 14 to 15 months to finish from the beginning to the tox production. And for the IND filing, might take some additional 8 months to complete the IND filing. The reason behind that is, in general, for biosimilar studies, we need to start with the analysis of the original drug, including its comprehensive profile. And in particular, it would require longer time for the upstream development to come up with a product that is statistically comparable to the quality profile of the originals. And on top of that, we need to purchase different batches of DS and DP for originals in order to appropriately design our process. So that is the reason why, in general, it takes about more than twice of the development timeline. So in general, it takes us about 6 to 8 months to finish the tox production for a monoclonal antibody. But it also depends on how fast can we achieve comparable profiles through the upstream optimization. So sometimes if the PTM or the charge variance is something difficult to be controlled, especially it's hard to be controlled through downstream, it might take even longer time for the development team to come up with the completed process.
Okay, thank you. And here is another question also related to the low PI. Could you please discuss the overall purification strategy for proteins with low PI, especially the capture step?
Okay, that is a very good question. And in general, as I mentioned, there were a number of challenges associated with proteins with low PI. So again, in particular, for the capture step, you need to take considerations a number of different factors. First, the stability of the protein. As I mentioned, it is always recommended to perform a stability study, right, before any chromatography steps to kind of narrow down the operation space for these proteins, because in our experience, in generally, low pH and low conductivity is quite unfavorable for those type of proteins, which will induce problems of aggregation, etc. And second is the selection of the chromatography step. Basically, as I mentioned, we typically recommend CEX step because it's avoided using a high salt concentration in the beginning as the high hydrophobic interaction chromatography, or it's kind of avoided some problems with AEX capture, which at the same time typically capture some pigments and other impurities, which will easily block the chromatography column. So carefully select the operation condition. If that could be applied in a CEX step, and you can possibly follow one of the examples I showed today, identify the best elution and potentially wash step with some tips to remove some HCPs and other impurities right in the beginning would be a good strategy. And third is always you need to consider the overall process for the protein. Sometimes even using a fourth chromatography step is necessary because of again some impurities and in particular HCPs to be removed. In those cases, we generally recommend hydrophobic interaction in between. This is typically quite effective to remove HCP, to bring HCP level down, and sometimes using mixed mode chromatography to help further polish the products.
Okay, thank you. Did you get any more questions from your Q&A panel?
No, I think that's all I have for now.
Okay, thank you. Okay, let's move to the next part. Now, please let me introduce the next speaker, Dr. Mengping Liu, Senior Scientist for Quality Study Department of GenScript ProBio. Dr. Liu leads the CMC Analytical Development Service for protein and antibody and provides technical leadership in new instrumental analytical development. She obtained her MS in pharmaceutical analysis from Sanya Sanity University and PhD in biomedical science from the University of Hong Kong. She published 5 first-author papers in well-known SCI journals and is dedicated to the quality study of biologics. In this talk, Dr. Liu will discuss ProBox solutions for analytical development. Now, I'm delighted to hand over to Dr. Liu. Dr. Liu, please.
Thank you for Jessica's introduction.
You're welcome. You can share your screen.
Yeah, sure. Can you see my screen?
Yeah, it's okay.
Oh, okay. Good afternoon. Hello, everyone. My name is Mengping Liu. It's my pleasure to present today's webinar to you, which is titled as Analytical Solutions for Biologics CMC Challenges. In today's topic, I gonna have a brief introduction of the ProBio analytical department and also introduce some solutions from the toolbox developed particularly for analytical and process development problems. So first, please allow me to give you a brief introduction of the ProBio analytical platform. Our analytical platform could provide multiple services for our clients, including creating the early drug development assessment, quality control in CMC, drug characterization, etc. And we have rich experience. What's going on? We have rich experience of developing over 200 analytical methods, analyzing 30 plus complex biologics. Our platform is equipped with advanced instruments from world-renowned vendors, just to guarantee the reliability and high throughput in CMC analysis. This table presents some analytical methods we developed in our platform. So basically, for each quality attributes, we have at least 2 methods, just to ensure the results are reliable. And we have alternatives in different scenarios. For example, for the titer measurement, we have developed ELISA, octet, and protein A HPLC methods. To guarantee the results can be applied for can well demonstrate the quality of the samples. Next, so as we know, the quality study for proteins is always challenging because these molecules have complex structures and bioactivity. Here, I listed some typical challenges from bispecific antibodies and recombinant proteins. For these 2 types of molecules, and they are usually expressed with a low titer, indicating sensitive methods should be developed for titer measurement. And the design of the bioactivity assay is also very challenging for both type of molecules. Because they have varied MOA, the mechanism of actions for different molecules. And for bispecific antibodies, they're usually expressed with various byproducts. For example, the mismatched homodimer, and they have very similar physicochemical properties to their main protein. So it's difficult to separate and identify these homodimer. And also that brings some problems for the purity analysis as well. Therefore, the recombinant proteins, and they are unstable and usually have complex PTM, the post-translational modification, and that leads to the challenge of analyzing the purity and PTM as well. So because of that, and we developed a toolbox, we named it as ProBox, and to address these problems I mentioned above. This is an overview of the analytical solutions in the ProBox. Next, I'm gonna demonstrate how we use this ProBox for the problem solving, particularly in the analytical development and our CMC process troubleshooting. First, I will give you several examples and to demonstrate the solutions we developed for the analytical development problems. This solution, the demonstration were driven by the real case studies in our projects. For the first type problem, low response signal in the non-reduced CSDS. This is a low pH stability study, and we found that the analyte shows a very low response signal in the non-reduced CSDS electropherogram after only 2 and 4 hours incubation under the low pH condition. That's unexpectedly. And we guess the high salt in the running buffer might cause this low signal because the analyte in the running buffer is supposed to be negatively charged after the SDS denaturation. When the buffer has a high concentration of the salt ions, and they would interfere with the charge of the analyte and leading to the change of, for example, the peak heights and the migration time. To verify the hypothesis, and then we exchange the samples into a low salt trace buffer. And you can see after buffer exchange, the signal of the analyte was back to normal and get a problem solved. And then second problem relates to the low resolution in the development of a high HPLC method. This is a IgG1 case. Yeah, we need to develop a high HPLC method for this molecule. And we first tried our platform method by using the column 1. Yeah, this is some information about this column. But we found that it's incapable of separating the oxidized products of IgG1. And then we switch to another column. Yeah, the column has a smaller particle size, and the ligand B as a solid phase material. And for this ligand, compared to the ligand A, it has a stronger hydrophobicity. And by using the column 2, we can see the resolution of the method was significantly increased. So basically, for this kind of problems, we can consider to using column with appropriate ligands and particle size. For the third problem is relevant to the poor quantification accuracy in the bioassay. Yeah, this is a competitive binding ELISA assay developed to test the binding affinity of the analyte anti-spike and to the target ACE2 receptor. We tried different binding formats. In format A, we immobilized the ACE2 target on the plate and then added the anti-spike and the competitor into the system. But we found that, this binding format leads to the inaccurate ELISA results. By seeing the binding curve, we can see the heel slope ratio is kind of out of the system stability test range. And that might be caused by the wrong binding formats. When the target is immobilized on the plate, and some active binding domains may not be able to be exposed to the antibody because of, for example, the steric hindrance effects. So we changed to another binding format, and here, we coated the spike, the competitor on the plate first, and then allow the analyte and the receptor freely interact to each other. With this modification, we can see the binding curve. Now the heel slope of the binding curve is right in the system stability test range, indicating the results using this format more reliable. Okay. And the next part, I'm gonna talk about some analytical solutions developed for CMC process development problems. So in the process development, and we often have molecular cleavage issues, particularly in the cell line development stage. And this case is related to a fusion protein, Fc-fused protein. It's very easy to degrade in very early process development stage. So here we need to develop a method to monitor the molecular degradation and to minimize the risk for downstream process studies. And for this protein, we listed possible fragments of the molecule. And here we developed Western blot assay to detect this cleavage process, because this assay is kind of very sensitive and specific for the molecule. And here, this is the antibodies we used for the assay development. Anti-Fc and anti-VHH, and that's specific to both the segments in the protein. Here, this is a later interpretation principles for this assay. Basically, for the full length of protein and these two segments, and because they have both Fc and VHH fragments, they can be detected by anti-Fc and anti-VHH antibody. And then for this VHH and for the individual VHH and Fc fragments, they can only be detected by their specific antibodies. We use this Western blot analyzing the proteins in the cell pool 165, and found that their fragments can be detected by both proteins. This is a possible fragments formats in this cell pool, indicating protein cleavage happened in this pool that cannot be used for further study. So for this type of problem, the molecular cleavage, we can consider to use, like, for example, the fragment-specific antibody-based Western blot to have an early idea of the cleavage in the cell line development. For second type of problems is to identify the PTM, post-translational modification. This is the IgG1 case. In this study, we found the main peak of the antibody was split in the high chromatogram. Based on our experience, we guess that might caused by the PTM from the incomplete, from the lysin and pyroglutamic acid cyclization. And so first, we use the specific enzyme to digest the split peak and to see whether it can eliminate the heterogeneity of the sample node. And then based on that, further to separate these two peaks and for further investigations by mass spectrometry. After the enzymatic digestion, we can see only a single peak was left in the high chromatogram, indicating our hypothesis is kind of confirmed. Later, we separated the peak 1 and peak 2 and the split peak for mass analysis. The mass data further confirmed that the PTM indeed was formed in this sample, including the pyroglutamic acid cyclization and the lysin RORs, particularly on the heavy chain of this antibody. And relative to the peak 2, the peak 1, the percentage of complete C-terminal lysin clipping and N-terminal pyroglutamic acid cyclization is much lower. And that leads to the early illusion of this peak in the high chromatography. So for this type of problems, we just suggested to use specific enzymatic digestion to have some initial ideas of the type of the PTM for further study. For the problem 3 is to investigate potency loss of the proteins. This case relates to IgG4 antibody. We found in the formulation process development, their antibody lost over 50% of potency after 2 weeks of storage at 40 Celsius. Usually, the loss of the potency can be caused by the molecular cleavage, degradation, or undesignable PTM. So we first performed the purity analysis of this molecule, and then followed by its further deeper investigations by mass spectrometry. This is a purity data of this sample after 2 weeks of storage. We can observe a fragment in front of the heavy chain in the reducing CSDS electrophoregram, and that's also observed in SDS page, indicating maybe the sample was degraded during the storage. And to further confirm that, we applied RP-LC mass. And this data suggests that the heavy chain indeed had degradation, and that was cleaved into 2 fragments, fragment 1 and 2. In addition, we observed oxidation in this heavy chain fragment, also the full length of heavy chain. So we guess the heavy chain degradation and oxidation may lead to the decreased potential for this antibody. So for this kind of problems in the process development, we suggested to investigate that by studying the molecular degradation or undesirable PTM. The problem 4 is relevant to the mesosample identification. Mesosamples are very common in bispecific antibodies. This case relates to astromatic bispecific antibodies. It's composed of 2 strands. By seeing its ICIF profile, we can tell that there's a possible homodimer formed in this antibody. And so we need to identify what it is for a quality control purpose. So here we first purify this PIC1 and for further study by mass spectrometry. But we met problems when doing the purification. When we just use a single affinity resin chromatography to do the purification, we can see both methods have failed and to obtain a highly purified protein. And then we tried another method by using a mixed mode chromatography strategy. It combines CX and Higg affinity resin, and finally we get a highly purified PIC1 for mass analysis. As identified by the mass spectrometry, and we can see that this PIC was indeed a homodimer that's composed of 2 strands. So for this type of mesosamples, they may have very similar physicochemical properties and to their target protein. It's very difficult to separate that using conventional methods. And so maybe you can try to use mixed mode chromatography to separate that for the study. Yeah, basically that's all for today's presentations. In summary, ProBio have a rich experience of analyzing difficult molecules, for example, the bispecific antibodies and recombinant proteins. And now we developed ProBox, and that could provide very effective solutions and for the problems derived from the process and the analytical development. Yeah. Thank you for your listening and welcome any questions.
Thank you for Dr. Liu's excellent presentation. If you have any questions, please kindly type it on the chat or Q&A panel. As you enter the questions, please also follow our LinkedIn account, GenScript ProBio. There will be more online sharing resources, and we will show them on our LinkedIn at the first time. Thank you. Okay, we received some questions from the audience. The first question is, do you identify the cleavage site of fusion protein? Do you develop the MAM, multi-attributes monitor method to identify the PTM of MAP?
Sorry again.
Okay, you can also check this question on the chat panel. Yeah, I'm not sure if you can see it.
I cannot see it.
Okay.
Sorry.
Do you identify the cleavage site of fusion protein? Do you develop the multi-attributes monitor method to identify the PTM of monoclonal antibody?
From my understanding, the question is to ask whether we have some strategies and to check the PTM of monoclonal antibodies. Identify a cleavage site. Okay. Okay. Thank you for this question. Yes. For the first part of the questions, yeah, indeed, we have experience of identify the cleavage sites for fusion protein, and yeah, in the case I presented earlier, is actually the cleavage sites from our experience is usually in the linker sites of this kind of protein, and yeah, so we usually should integrate it we should integrate the mass spectrometry to analyze its molecular weight and sequence coverage and for the identification. And for the second part of question, MAM, sorry, we don't have this method and for the identify the PTM of MAP. Yeah, hopefully, yeah, we'll consider to use that in future.
Okay,
work.
thank you. Okay, and the next question is, do you have in silico methods to identify the possible PTM from sequence, like demidation or oxidation?
Oh yeah, thank you for this question. Okay, yes, indeed we have developed several in silico methods and for the to identify the possible PTM. Yeah, we have expertise in that, and if you want to learn more for the silico methods, and we will invite our experts from the mass spectrometry platform, and they can give you more information for that.
Okay, thank you. And the next question is, how ProBio analytical platform supports process units in CMC activities?
Okay. Thank you for this question again. Yes, our analytical department works closely with the process team, and yeah, many support them in three aspects. And firstly, we monitor the quality of the samples closely by performing in-process tests, and yeah, that could give some there are some give the process team some guidance for further work, and we will report issues and once observed, and then to minimize the CMC development risk. Secondly, we provide analytical data, for the process team to develop, for example, the predictive models of processes to facilitate the scale up and the technology transfer. Thirdly, we have defined the quality attributes and the specifications for the product release. So we are highly experienced in developing the analytical methods fitting for purpose, and have strong problem-solving abilities. And this greatly accelerates in the process development of our clients.
Okay, thank you for your answer. And the next question is, bio-specific antibodies often generate misassembles with similar PI or size. What solutions you may use to separate and identify these by products?
Okay, thank you for the question. The very challenging misassembles from bispecific antibodies is mismatched homodimer. Yeah, as Dr. Lee and I mentioned earlier. For the homodimers with, for example, the similar PI but different size, and we suggest using, for example, the SDS page, SEC, HPLC, and CSDS to analyze, because they have a higher resolution to size variance. And for homodimers with a similar size but different PI values, and then we can consider using ICIF and IEX. These methods, they are more sensitive to the charge variance. And then for the homodimers and with similar size and the PI, and then the higher resolution methods, for example, the mass spectrometry, should be used for analysis.
Thank you. The next question is, what is your strategy for successful technology transfer, including analytical methods?
Oh, okay. For this question, from the analytical part, I would say for method transfer, with the first strictly follow the guidelines from the pharmacopoeia. And before the method transfer, we would discuss the transfer protocol with our clients, and to ensure their analytical method transfer can be initiated properly. And in this protocol, we will define, for example, the scope, duties, materials, equipment, acceptable specifications, and something like that. And just make sure both sides and can have a clear idea of what to do next. And also, all analysts involved in the transfer work will be trained on the test method. And if ProBio is the transferring unit, and then we could provide on-site trainings and if needed. And also timeline is critical. And the timeline of the method transfer will highly align with the process units, and for better coordination. And we are very open to any discussions, and when the transfer needs troubleshooting or negotiations.
Okay, thank you. I think this question is about tech transfer. If Mr. Zhang or Dr. Lee has more thoughts about this question, you can also talk freely.
Oh, for the technical transfer, for ProBio, we actually have a very comprehensive document regarding the technical transfer problems. The project will start with the project initiations, and responsible people and the project group will form together. After that, there will be a document transfer, following with technical and compliance evaluation. These technical and compliance evaluation, including all the different materials and analytical methods, formulation, process, equipment, and in-process control. All of these will have a gap analysis and for risk assessment. After the risks, after all this variation passes through the risk assessment model, there will be high risk factors will be further evaluated and further verified in ProBio. Technical transfer plan will be finalized after this step, and some of the minor process development, some of the technical transfer activity will start it. After that is process validation and a technical transfer report. So as I said, it's a comprehensive thinking and activity surrounding the entire technology transfer process. It's not an action, but a comprehensive project that have to be coordinated by different groups, including process development, MSET, manufacturing, and of course, analytical development group.
Okay, thank you.
Yep.
Okay, seems no more questions. That's all we have for today. If you have any more questions, please contact us through our official email. Let me join others in thanking Dr. Li, Mr. Zhang, and Dr. Liu for their time today. Thank you. Also, thank you for your attending today's online symposium. Look forward to meeting you again soon. I'll send the recording of this symposium to all registrations through email next week. You can check the recording next time. Thank you.